Good day, everyone. For today's topic, we will be discussing the toxic response of the nervous system. Review on the physiology of the organ system and function. The nervous system is classified into two types, the peripheral nervous system and the central nervous system. The PNS consists of cranial nerves, spinal nerves, and ganglia, while the CNS consists of the brain and the spinal cord. Overview of the nervous system. Several generalities that allow a basic understanding of the actions of neurotoxicants include the privileged status of the nervous system with the maintenance of biochemical barrier between the brain and the blood, the importance of the high energy requirements of the brain, the spatial extensions of the nervous system as long cellular processes, and the requirements of cells with such a complex geometry, the maintenance of an environment rich in lipids, the transmission of information across extracellular space at the synapse, the distances over which electrical impulses must be transmitted, coordinated, and integrated, and the development and regenerative patterns of the nervous system. The nervous system is protected from the adverse effects of many potential toxicants by an anatomical barrier between the blood and the brain, which is referred to as the blood-brain barrier. It is a layer of specialized endothelial cells around the brain which prevents toxic substances released in the blood from entering the brain. The blood-brain barrier contains xenobiotic transporters that transport some xenobiotics that have diffused through endothelial cells back into the blood. These xenobiotic transporters are the ones responsible for the uptake of some chemicals into cells and are very important for the export of chemicals out of the cell. If not actively transported into the brain, the penetration of toxicants or their metabolites is largely related to their lipid solubility and to their ability to pass through the plasma membranes of cells forming the barrier. However, spinal ganglia, autonomic ganglia, and small number of other sites within the brain are not all protected by the blood-tissue barriers. This discontinuity of the barrier is the basis for the selective neurotoxicity of some compounds. This illustration is a schematic diagram of the blood-brain barrier. In systemic capillaries, it has intracellular gap or what is called fenestrations, which allows the passage of molecules that cannot cross the endothelial cells and where there is more abundant pinocytosis. While in brain capillaries, it has tight junction and lacks pinocytosis, which limits the transport of compounds with active transport mechanism to be able to pass through. Neurons use energy in order to maintain proper ion gradient, which is why it makes it highly dependent on aerobic metabolism. The brain is extremely sensitive to even brief interruptions in the supply of oxygen or glucose. Exposure to toxicants that inhibit aerobic respiration like cyanide, or to conditions that produce hypoxia or being deprived of adequate oxygen will lead to early signs of neuronal dysfunction. The damage to the nervous system under these conditions is a combination of direct toxic effects on neurons and secondary damage from systemic hypoxia or ischemia. Axonal transport. Impulses are conducted over great distances at rapid speed, providing information about the environment to the organism in a coordinated manner that allows an organized response to be carried out at a specific site. Axonal transport moves protein products from the cell body to the appropriate site in the axon. Fast axonal transport carries a large number of proteins from their site of synthesis in the cell body into the axon. Axonal degeneration. When the neuronal cell body has been lethally injured, it degenerates in a process called neuronopathy. This is characterized by the loss of the cell body and all of its processes with no potential for regeneration. However, if ang injury is nasa level ng axon, may chance pa na mag-degenerate while yung neuronal cell body mag-continue mag-survive, making it possible to recover from the toxic injury. The result of axotomy or transection of an axon is that the distal axon is destined to degenerate, a process known as axonal degeneration, which is unique to the nervous system. The cell body of the neuron responds to the axotomy as well as undergoes a process of chromatolysis. In the peripheral nervous system, Schwann cells respond to loss of axons by decreasing synthesis of myelin lipids, downregulating genes encoding myelin proteins, and differentiating to a pre-myelinating mitotic Schwann cells phenotype. The proliferating Schwann cells create a tubular structure around the axon, which is termed as the band of Bungner, providing physical guidance for regenerating axons. The main difference that exists between the axonal degeneration in the central nervous system and the peripheral nervous system is that in peripheral axons, it can still regenerate, whereas the central axons cannot. The main factors contributing to the inability of the central nervous system to regenerate includes inhibitory factors secreted by oligodendrocytes, astrocyte scarring, and glial interference. Myelin formation and maintenance. Myelin is formed in the central nervous system by oligodendrocytes and in the peripheral nervous system by Schwann cells. Both of these cell types form concentric layers of the lipid-rich myelin with the progressive wrapping of their cytoplasmic processes around the axon in successive loops. The process of myelination begins when a myelinating cell, which can either be a Schwann cell or an oligodendrocyte, encircles an axon. Upon the enclosure of the axon, it then proceeds to the formation of progressive wrapping of multiple layers of the myelinating cell around the axon. The intracellular space is then compressed to form the major dense line of myelin, and the extracellular space is compressed to form the intraperiod line. Achieved in the nervous system through the help of synapse. Neurotransmitters released from one neuron act as the first messenger. Binding of the transmitter to the postsynaptic receptor is followed by the modulation of an ion channel or activation of a second messenger system, leading to changes in the responding cell. There are various therapeutic drugs and toxic compounds that can greatly affect the process of neurotransmission. Neurotoxicity expresses itself in terms of altered conduction and propagation of nerve impulses, and changes in functions such as behavior, performance, and conditioning. These chemicals act on neurotransmission, lead to interrupting the transmission of impulses, blocking or accentuating transsynaptic communication, blocking reuptake of neurotransmitters or precursors, or interfering with second messenger systems. The primary function of the nervous system is to collect a multitude of sensory information, process, interpret and integrate that information, and initiate appropriate responses throughout the body.